So today we will be discussing a process of acquired immunity called a T cell dependent B cell activation. So down here's the title, T cell dependent B cell activation. And the overarching idea is that we're turning a B cell, one of these guys, a B cell, into a plasma cell. And plasma cells have the ability to produce antibodies. And antibodies are these structures that have unique binding domains or regions that are able to bind with a surface component of some bacteria or a virus or a fungi or whatever cell is not considered a self cell. But under unique circumstances, sometimes they can bind with self cells and cause an autoimmune reaction, where now innate immune cells are recognizing that antibodies are bound to a self cell. Therefore, they're starting to attack self cells. So for example, these antibodies, let's say this is our antibody right here, will have two regions right here and right here that can bind with the surface component of bacteria. So imagine these little black guys are bacteria. This red thing is my antibody. And so this region right here is called the Fc region of an antibody. And innate immune cells, like macrophages or neutrophils, let's say this is a macrophage, have receptors on their surface that can recognize this region right here. And that's important because when I have an antibody, that antibody has a unique binding domain that can bind with this bacteria right here. And if a macrophage can recognize this antibody is bound to something, I can't necessarily recognize what that is. It can then consume this antibody and whatever it's bound to. So these antibodies are able to bind with a bacteria that I couldn't recognize before using my innate immune system, which allows me to consume whatever that antibody is bound to, enhancing the ability of my innate immune system, like my macrophages and my neutrophils, to recognize cells or fragments of cells or whatever invader or non-self cell is present that I couldn't necessarily recognize before. So this process of producing antibodies is what we'll discuss. And these antibodies are produced by a cell called a plasma cell. And plasma cells are formed by B cells. They're formed from B cells. B cells will eventually become plasma cells. And so this process is how do we rely on a T cell, a helper T cell, to help a B cell become a plasma cell so that I can produce antibodies that can bind with a bacteria that my innate immune system couldn't originally recognize. But now I can have innate immune cells, imagine this is our macrophage, that can come over and recognize, I see this antibody's bound to something. I can't recognize what that something is, but now I can because I can recognize antibodies. So this starts up in the skin usually. So this is a story starting in the skin. It could be in the gut. It could be in the lung mucosa. It could be usually on any sort of epithelial barrier. But this process in particular is happening in the skin. And so these cells called dendritic cells are positioned throughout the skin within kind of the interstitial space of the skin, so the watery environment, not in your epidermis, but kind of like your dermis and your hypodermis. And these dendritic cells are called Langerhans dendritic cells. But the idea is that these guys are positioned kind of all over the skin. They're monitoring the barrier between the outside environment and the inside environment. And they're kind of positioned like sentinels, where they have the role of detecting that something's wrong in the local environment. So let's say there's some molecules that are toxic, or I have some invading bacteria. Let's say this is my bacteria right here, and that bacteria has these little surface components that are unique to that bacteria. This dendritic cell can recognize something's up in the environment. I'm recognizing that toxins are floating around, inflammatory signal molecules called cytokines are floating around. And this dendritic cell can undergo a process called macropinocytosis, where it goes and it eats part of the environment. It just consumes part of the environment, but part of that environment is fluid. So macropinocytosis involves the consumption of fluid, And it's kind of a random process. So it's like it's randomly detecting something's wrong in the environment. Therefore, I'm consuming part of the environment. I'm bringing in whatever's in that fluid and I'm breaking it down. So this dendritic cell reaches its arms out into the environment, grabs that little bacteria, pulls it into the cell. And this is not mediated by a receptor. So it's kind of just, I'm grabbing whatever's there. If I get lucky and I grab something that's foreign, I bring it into the cell. So this is the little foreign object. It's now within a membrane within the cell. And this whole space right here would be considered the cytoplasm of the dendritic cell. So this little bacteria, or whatever it is, it could be a virus, it could be a fungi, it could be a fragment of our own cells, is considered to be in a space that's inside the cell. So this is inside the dendritic cell, but it's outside of the cytoplasm. So this is a unique environment for this little bacteria to be residing in. And the next step for this dendritic cell is to take an organelle called the lysosome and fuse the lysosome with this little vesicle. And the lysosome will have enzymes within it that can break down proteins, denature proteins, which are amino acids that are all strung together, but they have kind of like this 3D shape. But if I break that all down and I unstring those amino acids, I'll have individual amino acids that are still bound by a peptide bond, so they're still connected. But it's where I have one amino acid, two amino acids, three amino acids, four amino acids, all connected in a linear strand. So I have this lysosome, it's breaking that little bacteria down and all the proteins that compose that bacteria, along with the membrane lipids, along with the cell wall. But important part is the peptides, the proteins. And it breaks it down into little individual fragments. So you can see over in this picture over here, it's broken it down into little peptide fragments. And then these dendritic cells are what are called antigen presenting cells. Abbreviated antigen presenting cells, or APCs. So antigen presenting cells are cells that have the ability of producing MHC2 molecules. And MHC2 molecules are major histocompatibility type 2 molecules. But fundamentally, they're a receptor that is expressed on the surface of the cell that holds peptides. So it holds the peptides that I broke down by the lysosome. So imagine that little blue guy is my peptide, where I have amino acids 1, 2, 3, 4. Usually they're a specific length, and you have to have certain amino acids present in order to be presented by this MHC2 molecule. But these dendritic cells can inherit genes from the mother and from the father. And typically there's 3 genes associated with the mother's chromosome and 3 with the father that can give rise to these MHC2 molecules. So on the surface of this dendritic cell, you can present upwards of 6 different kinds of MHC2 molecules that can present a wide variety of different peptides. And so this dendritic cell will present on its surface, I'll draw this out, on its surface a MHC2 molecule, this is my MHC2, with a foreign peptide. And that foreign peptide is foreign because it was originally taken in from the environment. And so this dendritic cell is presenting that foreign peptide on the surface. It's then migrating from the skin, from the peripheral epithelium, it could be from the gut, from whatever, towards secondary lymphoid tissue. And it's usually following a gradient of chemicals that are released by the epithelial cells that actually form the lymphoid tissue. So it's following a gradient of signal molecules that are telling it to move towards the secondary lymphoid tissue, where it's then hanging out and it's presenting an MHC2 molecule right here with the foreign peptide that we got from that bacteria that was originally outside the cell. And then this dendritic cell will also present a B7 danger signal. And the B7 danger signal is functionally significant because it tells these naive T cells to come try their fit and see if their T cell receptor that's on the surface of a naive T cell can come over and bind with the foreign peptide. So naive T cells are cells that are not necessarily mature yet, And each of these naive T cells has a pretty unique T cell receptor, which means that they have the T cell receptor, but it has a unique shape that can bind with foreign peptides that are presented by MHC2 molecules. So I've got a bunch of different kinds of naive T cells. Let's say I have a few more in the environment. This is another guy. This is another guy. And they all have T cell receptors. This is a T cell receptor. This is a T cell receptor on the surface of the cell. Each one of those T cell receptors, this one, this one, this one, has a unique shape. And depending on that unique shape, it allows that T cell receptor to bind with the foreign peptide. So let's say we got lucky and this naive T cell had a T cell receptor that could bind with the foreign peptide. You may ask, what is actually binding to the MHC2 molecule? Because this binds with the foreign peptide. What binds with MHC2? So I'll scroll over, and there's another protein on the surface of a naive T cell called the CD4 protein. So these naive T cells are often called CD4 T cells or T4 T cells because they have a CD4 protein positioned on the surface that binds directly with the MHC2 molecule. So we have the T cell receptor binding with the foreign peptide. We have CD4 binding with MHC2. And then this B7 danger signal is what's called a co-stimulatory molecule. So it's on the surface of this dendritic cell along with the MHC2. And it coaxes these naive T cells to come try their fit, see if their T cell receptor can fit the foreign peptide. If it doesn't fit, nothing happens. But if this B7 danger signal is not present, this naive T cell, even if the T cell receptor binds, cannot actually undergo clonal expansion and differentiation, even though everything fits. So this B7 danger signal is very important because if it's not present, these naive T cells will then go into a state called anergy, ANERGY, which is where nothing really happens. They're kind of in a quiescent state. And so if everything fits, this naive CD4 T cell, because it has a CD4 protein, will then undergo clonal expansion. So I'm making a bunch of copies of this guy. And then eventual differentiation. So I'm turning each one of those copies into a specific differentiated cell type. So these cells are unique cells that have a T cell receptor that's the exact same shape as this one. So I've functionally made a bunch of copies of these helper T cells that all have T cell receptors that can fit this little foreign peptide right here. And once I form a population of these helper T cells, typically follicular helper T cells, these follicular helper T cells have a T cell receptor that can bind with a B cell that's also presenting an MHC2 molecule, and they can help that B cell become a plasma cell so that I can produce antibodies that can bind with a surface component of the bacteria and allow my innate immune system cells to recognize that that antibody is bound to something I can't recognize. Therefore, I'm going to consume whatever it's bound to and neutralize the bacterial or the viral or the fungal invasion. For this example, we're kind of just focusing on bacteria, but it could be related to whatever sort of microbe or fragment of a cell or toxin or whatever is present in the local environment within the body. But there's also antibodies that can be produced and released into spaces that are inside the body but outside of an epithelium. Some of those spaces could include the gut or within the space within the lungs, so the lung mucosa. They could be released into nasal passages, wherever. So this is an example of it being released inside the body, so within circulation typically. But they can be released in a wide variety of locations. They can be excreted or secreted. And so we have to talk about, we form this population of follicular helper T cells that each have a T cell receptor, and they help the B cell become a plasma cell. But how did I get a B cell? Where are they located? And what's their functional significance? So if I go up to the original problem, I had an invasion of bacteria Some of that bacteria interacted with dendritic cells within the skin, but let's say some of the bacteria is flushed into the lymphatic system. And if some of the bacteria is flushed into the lymphatic system, into lymph vessels and eventually lymph nodes, which are considered secondary lymphoid tissue, they can interact with cells called naive B cells. These guys are called naive B cells. They're not quite mature B cells yet, and naive B cells will have what are called B cell receptors on their surface. And B cell receptors are just antibodies. And so one naive B cell will have completely different B cell receptors on their surface as another naive B cell. So imagine these are two naive B cells located in the same region. This is a naive one, this one's a naive one. Both are naive, but these B cell receptors are completely different from these ones. They can bind to different surface components of the bacteria. So you have a wide variety of naive B cells. If you get lucky and you have a naive B cell with a B cell receptor that can bind with the surface component of this bacteria, this naive B cell can bring that little bacteria in that's bound to that B cell receptor through a process called receptor-mediated endocytosis, because we're using a receptor to bring in that bacteria through the process of endocytosis, bringing it within the cell. And then we take that little bacteria and we put it in a space that's within the cell, but outside of the cytoplasm. So this is a little vesicle, let's say. This is the little bacteria within the vesicle. This is the vesicle. And that B cell will also have a lysosome that will go over and fuse with the vesicle. It'll release a bunch of enzymes that start degrading that bacteria, denaturing the proteins that make up that bacteria into little peptide fragments. And so that B cell is also considered an antigen presenting cell because it has the capacity of producing an MHC2 molecule. So MHC2. And that B cell will produce an MHC2 molecule. It'll then fuse that MHC2 molecule right here with one of the foreign peptides that I broke down from that bacteria that I got from outside of the cell. And that MHC2 molecule will fuse, and then it'll be presented on the surface of this B cell. So right here, highlighted, is the MHC2 molecule that's presenting the foreign peptide in red. And it's presenting it to follicular helper T cells that have T cell receptors that can fit the foreign peptide presented by MHC2. And these follicular helper T cells, also on their surface, still have the CD4 protein that can bind with the MHC2 molecule. And then, similar to what we saw with the danger signal, this B cell will co-present or present a co-stimulatory molecule called CD40 that can bind with a CD40 ligand, which kind of just coaxes this follicular helper T cell to come try its fit, see if its T cell receptor can fit the foreign peptide. And if everything fits, it allows this B cell to differentiate or to first undergo clonal expansion. So I'm making a bunch of copies of this B cell, a bunch of different copies. And then each one of those copies is differentiating or turning into a plasma cell that has the ability of producing a bunch of antibodies within the cell and then releasing them into a space that's outside of the cell, typically interstitial space or circulation. And there's different kinds of antibodies. Here depicted is the IgM antibody, or the immunoglobulin M antibody, which is a pentamer where you have 5 different antibodies all bound together. And these are typically released into circulation, and they can go and bind with bacteria or fungi or viruses or whatever surface component I originally recognized up here. And they can bind with that bacteria and help my innate immune system recognize something that I couldn't recognize before. So overall, this is a picture of how did we form a population of helper T cells? How did we form a mature B cell? And once I had helper T cells, specifically follicular helper T cells, how did those follicular helper T cells help a B cell become a plasma cell that could produce antibodies to help my innate immune system, like my macrophages or my neutrophils, recognize a bacteria or a virus or some surface component of something that's foreign that I couldn't necessarily recognize before. Thanks.